Good morning, ma'am. My topic for the seminar is role of nuclear transplantation in development, example Acetabularia. What is nuclear transplantation? Nuclear transplantation is a method in which the nucleus of a donor cell is relocated to a target cell that has had its nucleus removed or enucleated. Nuclear transplantation has allowed experimental embryologists to manipulate the development of an organism and to study the potential of the nucleus to direct development. Somatic cell has the ability to generate a complete organism, and the nucleus of a somatic cell must be reprogrammed if it is to participate in normal development with an enucleated egg. Nuclear transplantation, as it was first called, was later referred to as somatic nuclear transfer or cloning. Nuclear transplantation experiments was initially performed on Acetabularia and then on amphibians. My topic is on Acetabularia. In 1930s, Hammerling took advantage of the unique features between two morphologically distinct species, that is Acetabularia mediterranea and Acetabularia crenulata, and conducted experiments with them. Acetabularia mediterranea has a smooth disc-shaped cap, while Acetabularia crenulata has a branched flower-like cap. Each Acetabularia cell is composed of three segments: foot or base, stalk and cap. Experiments conducted by Hammerling were exchanging nuclei between two species of Acetabularia. He grafted the stalk of one species of Acetabularia onto the foot of another and fractionated young Acetabularia into several parts. Experiment 1. Exchanging nuclei between two species of Acetabularia. Acetabularia mediterranea and Acetabularia crenulata have two different cap structures. Hammerling found out that when he transferred the nucleus from one species into the stalk of another species, the newly formed cap eventually assumed the form associated with the donor nucleus. Thus, the nucleus was seen to control Acetabularia development. The formation of a cap is a complex morphogenic event involving the synthesis of numerous proteins which must be accumulated in a certain portion of a cell and then assembled into complex species-specific structures. The transplanted nucleus does indeed direct the synthesis of species-specific cap, but it takes several weeks to do so. Thus, Hammerling experiment with single-cell Acetabularia showed that the nucleus of a cell contains the genetic information that directs cellular development. Experiment 2. Grafted the stalk of one species of Acetabularia onto the foot of another. Hammerling grafted the stalk of one species of Acetabularia onto the foot of another species. In all cases, the cap that eventually developed on the grafted cell matched the species of foot rather than that of the stalk. In this experiment, the cap that is allowed to grow on the grafted stalk looks like the base species one, that is Acetabularia mediterranea. The experiment shows that the base is responsible for the type of cap that grows. Thus, nucleus that contains genetic information is in the base, so the nucleus directs cellular development. Experiment 3. Fractionated young Acetabularia into parts. Hammerling fractionated Acetabularia mediterranea into three parts: apical tip of stalk, central portion of stalk, rhizode with nucleus. And his observations were: apical tip of stalk showed cap and stalk regeneration, and central portion of stalk showed no generation, and rhizode with nucleus showed total regeneration. Hammerling knew that the portion with the nucleus would be regenerated, but apical tip of stalk regeneration surprised him. Hence, as Hammerling concluded nearly 30 years ago, before the existence of mRNA was known, that the instruction of cap formation originated in the nucleus and somehow stored in a dormant form near the tip of stalk. Many years later, researchers explained that the cap regeneration without nucleus indicates that nuclear information had previously been transferred to the cytoplasm and persisted there, at least through the time required for the formation of new cap. The information in the cytoplasm for regenerating the cap takes the form of mRNA.